So the 30th question is about sickle cell anemia. So sickle cell anemia is an example of pleiotropy. Answer is answer number 4. When we talk about pleiotropy, the basic idea is that when one gene affects multiple traits. So that is not the definition. There is a proper definition in the resource book, but the general idea is because of a problem in one gene, many other features are affected. So if we consider this sickle cell anemia, in people with sickle cell anemia, their red blood cells are shaped like sickles. So they have sickle shaped red blood cells. Normal red blood cells are disc shaped by concave, but these people, their red blood cells are sickle shaped. So because of these sickle shaped red blood cells, they can have many other problems like renal failure, heart failure, thrombosis, like that. So you can see because of a problem in one gene, other traits are affected. So that is the basic idea of pleiotropy. Then the other example for pleiotropy is cystic fibrosis. So in cystic fibrosis, what happens is the mucus is stickier and thicker than normal, and because of that problem, many other problems can be created like lung infections, infertility, and respiratory failures, like that. So that is the basic idea of pleiotropy. Then the 31st question. So it's about this cross, capital R, simple r into capital R, simple r. So if you draw the cross and consider the final, the first F1 generation, you can see the probability of getting simple r and simple r is 1 over 4. So there are 4 offspring. The four probabilities are there. From these 4 different probabilities, there is only one probability for getting simple r and simple r. So because of that, the first statement is wrong. Then the second statement says this is a dihybrid cross because 2 alleles are involved. That is completely wrong. This is a monohybrid cross. So in monohybrid crosses, one character is involved, and this one character is represented by 2 alleles. So in dihybrid crosses, 2 characters are involved, and for each character there will be 2 alleles. So in dihybrid crosses, there should be 4 different types of alleles. So because of that, the second statement is also wrong. Then the third statement says according to Mendelian inheritance, if we crossbreed the F1 generation, we have to get 9 over 16 probability for the dominant phenotype in the F2 generation. That is wrong. It is there is no rule like that. Then the fourth statement, it says if we get 1:2:1 ratio of phenotypes in the F2 generation, it is because of codominance. That is correct. So we have a section called non-Mendelian inheritance. That means when these genetic crosses, they don't follow the normal rules that Mendel proposed. So under non-Mendelian inheritance, we learn about incomplete dominance, codominance, polyallelism, and epistasis. So we have to remember the ratios. That means if we consider incomplete dominance, you have to know in the F2 generation, the ratio is 1:2:1. Then in codominance also, in the F2 generation, the ratio is 1:2:1. In polyallelism, in the F2 generation, the ratio is 1:1:1:1. Then if we consider dominant epistasis in the F1 generation, the ratio is 13:3. If we consider recessive epistasis in the F1 generation, the ratio is 9:7. So we have to remember those ratios. So by that we can see 1:2:1 is codominant. So fourth one is the correct answer. Then the fifth statement is wrong. They haven't given enough information to tell whether these are linked or not. So the fifth statement is wrong. Answer is answer number 4.